The aim of upstream analysis is to identify which upstream regulators may be activated or inhibited to drive the differential expression or phosphorylation changes you see in your dataset. This can help you better understand how regulatory cascades control your experimental system. IPA users often use this section for identifying key regulators contributing to disease pathogenesis, drug targets and mechanism of action, as well as CRISPR and RNAi assay design. In the case of a gene expression dataset, upstream regulators are not only transcription factors, they can be any molecule known to impact the expression of other molecules. And in the case of phosphorylation analysis, IPA goes beyond identifying just kinases and phosphatases as upstream regulators and includes any molecule known to impact the phosphorylation level of other proteins. Here's an example of a top upstream regulator prediction. IPA has predicted that PPARA, a ligand-dependent nuclear receptor, has been activated, and that activation has causally led to the up and down regulation of these genes from the dataset. The default view is to display the regulator in the center of a network surrounded by its targets. In this case, the genes that it is known to regulate based on information curated from the literature. The table is arranged so that the upstream regulators with the most significant p-values of overlap with its targets are shown at the top. If an upstream regulator is in the dataset, you can see the expression, phosphorylation, or variant measurement values in this column. Empty cells within this column represent upstream regulators that are not in your dataset. These regulators are included in the results because information from the knowledge base indicates that they target a significant set of analyzed molecules in your dataset. The regulator is often not itself differentially expressed or phosphorylated, yet its influence can be predicted by the changes seen in the dataset. For example, post-translational modification, loss of an inhibitory protein, or localization to the nucleus may be responsible for the molecular changes observed in your experiment. These columns contain the predictions of activation and their associated z-scores. Z-scores greater than or equal to 2 represent statistically significant predictions of activation, while z-scores less than or equal to -2 represent statistically significant predictions of inhibition. The target molecules and dataset column shows the molecules in your analyzed dataset that are targeted by an upstream regulator. When you click on this hyperlink, a new table appears, which shows more specifics about why an upstream regulator was given an activity prediction. You can view additional information about the upstream regulators by selecting the Customize Table option. Here, you can add the Benjamini-Hochberg corrected p-values, bias-corrected z-scores, and more to the table. Earlier, I showed you an upstream regulator network. You can see this type of result when you select the Display as Network button. This option provides a graphical network showing how a regulator interacts with its downstream targets. The legend on the right side of the screen shows a description of the color coding used in the network. The final column is Mechanistic Network. This function allows you to view a network of multiple upstream regulators that interact with a particular upstream regulator and its target molecules. You can also use the Causal Network Analysis feature to identify master upstream regulators. When setting up your analysis, you have the option to evaluate causal networks that contain molecules that are known to influence specific molecules, diseases, or biological processes. For more information about upstream analysis, you can go to the Help menu to access our online help manual or watch our video tutorials and webinars.